Welcome to our webinar about best practices and useful tips for RNA sequencing, hosted by Lexogen and BigOmics. My name is Yvonne, and I'm Head of Product Management at Lexogen. With me today is Axel Martinelli, Head of Biology at BigOmics Analytics. After a short introduction, we will dive right into the best practices and useful tips for RNA-Seq. We will follow by giving a short overview of the most common methods, namely whole transcriptome sequencing and mRNA-Seq for expression profiling, and show you how these two methods and datasets compare. Axel will show you around the Omics Playground, a cloud-based tool to facilitate and accelerate data analysis and insights on a practical example comparing 3' mRNA-Seq and whole transcriptome datasets for differential expression, gene ontology, and pathway analysis. Before closing, we will focus on the power of replicates and controls for your RNA-Seq experiments. We will start with a brief introduction. At Lexogen, we are a group of scientists with a passion for RNA and the aim to serve the scientific community and advance research progress. Lexogen was founded in 2007 and is headquartered in Vienna, Austria, with Lexogen Inc. operating out of New Hampshire in the US. Lexogen is focused on innovative solutions for RNA research and transcriptomics and is ISO 9001 and 13485 certified. We provide products, for example, in form of kits for you to use in the lab, with a focus on high-quality, cost-efficient RNA-Seq solutions. Our services provides customizable NGS workflows, including various RNA-Seq solutions, such as expression profiling, drug screening, whole transcriptome analysis, small RNA-Seq and biomarker discovery, as well as DNA sequencing, sequencing only, and data analysis. We also partner with other labs and companies like BigOmics Analytics to build our new technologies and make NGS workflows more accessible for everyone. BigOmics Analytics is a Swiss company that expedites tertiary data analysis by providing a centralized platform solution for scientists to efficiently scale their omics data analysis and obtain reproducible results. Through the collaborative analysis platform, Omics Playground, researchers can interactively share data and insights, fostering efficient collaboration. Omics Playground offers RNA-Seq and proteomics data analysis solutions. And today, Axel will focus on the RNA-Seq capability and walk us through some interesting features and insight for transcriptomics experiments. Now we want to focus on today's topic: experimental planning, best practices and tips for successful RNA-Seq experiments. Before we start, there's a question for you. What is your major challenge in setting up an RNA-Seq experiment? Interestingly, many researchers directly dive into the experiment itself, setting it up in a lab, methods, protocols, or handling related topics. In reality, the first hurdle is experimental planning. Experimental planning is the most crucial step in the RNA-Seq workflow, and when done right, it will set you and your project up for success. There are several considerations that require meticulous planning and a lot of thought before picking up a pipette. The major aspects revolve around experimental design, sequencing design, and quality control. Already at this step, you should consider which model system is suited for your specific aim, the limitation and requirements of your sample type of choice, the conditions you need to compare, replicates and controls, how you want to sequence your samples, which library prep you should use, and what you will need for data analysis, and how you can ensure reproducibility. Going a bit more in depth here. The organism and sample type you need to work with may already dictate which wet lab workflow you need to use. For example, which extraction and library prep protocols are suitable and which ones are not. The same goes for the conditions themselves. You need to ensure to choose suitable controls. For example, include a wild type and a rescue if you want to ensure that the effects you're seeing in a mutant are actually caused by the mutation you're analyzing. How many samples you're including and the number of replicates have a considerable impact on the conclusions, and they are key for biologically sound interpretations. We will come back to this part a little bit later on. Similarly, controls are extremely valuable to assess the quality of your data, how accurate your analysis operates, and they are key to detect any biases in your workflow. The sequencing mode depends on the type of analysis you want to do or the library prep you used. And quality and reproducibility are important for large-scale screens, experiments across different sites, or over a long period of time. But also considering the increasing lack of reproducible and scientific studies, they are a major challenge in science and specifically for high throughput studies that use NGS. Now that we've had a glimpse into the complexity of the considerations for RNA-Seq planning, let's take a step back and start with the basics. The first and most important question to answer is the purpose of your RNA-Seq experiment. Which problems are you trying to solve? What is your aim? Your research question drives the whole experimental design. It determines the type of data you need to solve your question, and it also impacts the sample type, which may impose a certain set of restrictions. Last but not least, considerations on how you can control your experiments and the analysis to draw valid conclusions are key. This includes everything from picking the right conditions to in-process controls or quality parameters that you need to check to ensure the integrity of your dataset before diving into the complete analysis. When you have your research question thoroughly defined, you will also have an idea which organism or model system you are working with. Though RNA is RNA, you need to treat plants differently from animals to extract it. So ensure you investigate suitable extraction kits and processing steps to make sure that your input RNA is of the best possible quality. Next, what type of RNA are you interested in? Messenger RNA, small RNA, non-coding or circular RNA, or all of it? Each type might require adaptations to the extraction method, pre-processing steps, or dictate choice of library prep. For example, if you're interested in small RNA, you need to pick an extraction kit that retains or even enriches these small RNA species. Up to this point, your organism or model system of choice may have specific requirements for the sample handling or the extraction method, like retaining small RNA, or being able to break open rigid cell walls or removing secondary metabolites. The cell type may also require additional gDNA removal steps. For example, blood contains 10 times more DNA than RNA, and bacteria often contain plasmids. So here, an additional DNA digest is very, very important. If you are interested in mRNA, or if you want to include non-coding RNAs, you will go either for mRNA selection or rRNA depletion. In-prep mRNA selection is also an option, especially when you're processing many samples, and sometimes you need to enrich targets after the library preparation is done. And the type of data matters. Do you need expression data, or do you need to see transcript isoforms? Are you interested in identifying targets of small RNAs, for example, or proteins? Or are you interested in kinetics, RNA half-life, synthesis, and decay rates? The questions if you need quantitative or qualitative data will also determine your choice of library prep method. provided your model system allows your choice. Basically, do you need to know if a gene is expressed and at which level, or do you require in-depth transcript information, for example, spliceoform and isoform expression information? Two commonly used methods are whole transcriptome sequencing, this is the standard, and 3' mRNA-Seq. Whole transcriptome sequencing, or WTS, is suitable for mRNA or total RNA-Seq. So total RNA, meaning mRNA and long non-coding and circular transcripts, while 3' mRNA-Seq only detects polyadenylated mRNAs. Therefore, 3' mRNA-Seq is dedicated to expression profiling. You can count one fragment per transcript for expression analysis, but whole transcriptome sequencing is more versatile and allows various different types of analysis. It provides qualitative data. While whole transcriptome sequencing requires complex data analysis steps to assemble the reads along the transcripts correctly, 3' mRNA-Seq accurately quantifies expression by straightforward read counting, which can considerably increase the speed of analysis and save computational power. Whole transcriptome sequencing covers the complete transcript and provides a uniform and even coverage. In contrast, 3' mRNA-Seq focuses reads on the 3' UTR and ultimate exon, which are efficiently covered during the sequencing experiments. Ultimately, 3' mRNA-Seq requires less computational resources, reduces the turnaround time for your data analysis, and provides accurate expression information through read counting. Whole transcriptome sequencing can resolve isoforms, but the data analysis is more complex and it requires more computational resources. However, it also provides a higher level of information. To resolve the underlying isoforms, whole transcriptome sequencing experiments routinely use more sequencing depth than the 3' method. Another aspect to consider when planning your experiment. While 3' mRNA-Seq relies on the availability of poly-A tails and is thereby limited to eukaryotes, whole transcriptome sequencing can be used for any sample or any organism. This table provides a quick overview of the different applications. 3' mRNA-Seq is dedicated to expression profiling. As the selection is done in prep, the input is total RNA without any pre-selection. This means a quick and highly scalable workflow. Due to the focus on the sequencing reads on the 3' end of the transcripts, a low sequencing depth of 3 to 5 million reads per sample is sufficient for most applications. Another point for scalability here. And all RNA qualities can be processed provided a canonical poly-A tail is present. So this method cannot be used for bacteria, for some viruses, and for RNA of interest that don't possess poly-A tails. The whole transcriptome pendant is mRNA-Seq, and it also delivers expression information on genes and transcripts, as well as isoform, alternative splicing, and fusion information. Due to the required poly-A selection upfront, only high-quality RNA can be successfully processed. For low-quality RNA, ribodepletion is recommended, leading us to total RNA sequencing, which covers additionally non-poly-A RNAs, such as long non-coding RNAs, circRNAs, SNO-RNAs, and it is also suitable for any species without poly-A tails. As the whole transcript is covered, higher sequencing depth is required, depending on the organism and application, typically between 25 and 100 million reads per sample. As a rule of thumb, the larger the genome and the lower the expressed RNA of interest, the higher is the required sequencing depth. As we have seen from the overview, both whole transcriptome and 3' mRNA-Seq can be used for gene expression profiling, and both methods are valid options for various sample types. The gene detection overlap shown on the left and the correlation plot on the right indicate that both methods deliver highly consistent and comparable results, though whole transcriptome sequencing detects more genes, which is expected. At this point, we want to go a bit more into detail and focus on how 3'-seq and whole transcriptome datasets compare. We already mentioned that both methods deliver comparable results for gene expression. Also, whole transcriptome, or short WTS, detects more genes or differentially expressed genes due to its higher diversity and depth. However, on a pathway level, the biological conclusion remains the same, regardless which prep is chosen. This makes 3'-seq ideal for scaling up. All the while, whole transcriptome sequencing offers higher resolution and is definitely a good choice to complement or to follow up your experiment once you want to go into more detail. So much to the theory. At this point, I'm handing over to Axel to look at the published dataset and see the comparison of 3' and whole transcriptome datasets and the Omics Playground in action.
So we are now going to take a look at the dataset using the Omics Playground platform. First of all, I'm going to focus on the comparison between whole transcript sequencing versus 3' mRNA sequencing. And here we can see the volcano plot of the up and down regulated genes. In the first plot here, you can see the WTS approach. And then in the second plot here, you can see the 3' mRNA-Seq approach. And you can clearly see that there are more genes identified using the WTS approach as the 3' mRNA-Seq approach, which is expected. And this we can further confirm by doing a Venn diagram where we intersect the two approaches. And we can see that there is obviously a larger number of differentially expressed genes in the WTS approach, which is shown here at the bottom in red, compared to the 3' mRNA-Seq approach at the top. But what you can see is that the vast majority of the genes that have been identified as differentially expressed using the 3' mRNA-Seq approach are conserved with the WTS data. And you can also see that there is a pretty good correlation between the two data sets. Now, the crucial aspect is when it comes to identifying differentially expressed gene sets. Now, if we do a traditional GSEA analysis, and we focus first of all on the WTS dataset, we can see that among the most upregulated gene sets, we see pathways associated to cholesterol biosynthesis. We can see different gene sets associated with immune responses, as well as the presence of changes in the circadian clock. And also here at the top, we can see a gene set associated with heme deficiency. Now, if we switch to the 3' mRNA-Seq data, and we do exactly the same, so we order them by most upregulated. So first of all, the heme deficiency gene set is conserved. We can also see the presence of various changes in inflammatory and immune responses. So we can see, for example, the same eosinophils, migration, and chemotaxis gene sets that were identified with the WTS data. We can also identify the presence of changes in the circadian clock, but we don't see any evidence of cholesterol biosynthesis alterations. Now, we can further enhance this analysis by looking at pathway enrichment analysis. And in this case, we are going to use the Wiki Pathways database. So again, if we look first of all at the WTS data, and we look at the list of the most upregulated pathways, we can see that again, we recognize cholesterol biosynthesis as being affected. We can also see evidence of changes in immune responses, and we can see changes in the circadian clock. Now, if we do the same with the 3' end data, we recognize, first of all, greater conservation of the top terms. We can finally identify cholesterol biosynthesis as being affected, as well as the presence of changes in immune responses, as we detected before. And so this indicates that by combining gene set enrichment analysis with pathway enrichment analysis, we can actually we can capture the changes, the majority of the changes that occur at the WTS level, also with the 3' mRNA-Seq data. So combining these two approaches is quite crucial to get a complete picture. Now, to finish the analysis, I'm going to focus on the importance of adding replicates in your data. So I'm going to look at the same data set, and I'm going to perform a gene set enrichment analysis, but now focusing on a data set where I only picked a random sample for the diet condition and the control condition versus the analysis where I included all the replicates. So here we see the same analysis that we had before, where we have all our replicates, and you can again see the same terms that we saw before. There is cholesterol changes, there is changes in immune and inflammatory responses, as well as changes in the circadian rhythm. Now, if we go to the data set where we only have one replicate, and again, we sort them by the most upregulated, we can see that basically the only terms that is conserved is the heme deficiency term that I discussed before, but now we cannot detect anything related to changes in cholesterol biosynthesis. We cannot detect any terms that are related to changes in immune responses or changes in the circadian rhythm. So while I understand that sometimes it's very difficult technically, or it can be very expensive to produce replicates for a given data set, this example highlights the fact that the absence of replicates can very often lead to missing most of the signals that are present in your experiment.
Thank you, Axel, for the demo and data insights. Before we follow up with replicates and controls, I'd like to quickly summarize Omics Playground. It enables significant time savings for data analysis and is compatible with both whole transcriptome and 3' mRNA-Seq datasets, as we've just seen. The platform also provides reliable conclusions, interactive visualization, and data interpretation support. So now, as 3'-seq and whole transcriptome sequencing compare extremely well at the level of the conclusion, which prep should you choose? Basically, you could choose whole transcriptome or total RNA-seq if you need a global view of all RNA types, information about alternative splicing, isoforms, or fusions, and if you work with highly degraded samples, samples that do not possess a poly-A tail, or when you work with organisms that are not very well annotated. You should choose 3' mRNA-Seq if you need accurate and cost-efficient expression quantification, if you need to process a high number of samples and scale up, if you require a short workflow and simple analysis, or need mRNA profiling from degraded samples and FFP material. The last topic for today focuses on the power of replicates and controls to reiterate what Axel has shown before. Replicates are extremely important for your experiment and conclusion. Not only do you miss a massive amount of information, the lack of replicates also leads to absolutely different conclusions. Moreover, most modern analysis tools for differential expression require at least 3 replicates per condition for reliable analysis. There are 2 main types of replicates: biological replicates that are derived from 3 different samples, be it animals or cells, cultures, or plants, or technical replicates, which use the same sample, but in 3 different preps to account for technical variation during the lab workflow by different operators, different environments, or during sequencing. Biological replicates measure the natural difference between individuals. If this natural variability is high, you should increase the number of replicates if this is possible, and also use technical replicates to reduce the noise as much as possible and to draw valid conclusions for the changes that you're actually interested in. A second measure to control your experiments and increase reliability is to include controls. SERVs are artificial spike-in variant controls that are added to the sample and processed through the same workflow. Only about 1% of total reads are then dedicated to the SERV controls. And even this little fraction is sufficient for the validation of RNA-seq workflows, assessing the performance and the limits of the given setup. SERVs are artificial sequences, and therefore they can be universally used for any sample type, any species, and with any sequencer. They allow validation and monitoring of RNA-seq workflows and assessing technical parameters such as bias, sensitivity, as well as the length representation and evaluation of functional aspects like isoform detection and quantification. This allows you to know the performance and the limits of your RNA-seq workflow and your data evaluation pipeline. Moreover, with spike-ins, you can evaluate the comparability of samples in experiments, but also between experiments. Therefore, you can evaluate the technical variability between samples and experiments before you're actually starting to investigate the biological variability. And with this, we are at our summary for today. A clearly defined hypothesis and objective guides the entire experimental process, from the planning through the lab and sequencing workflows and the data analysis. Optimal sample size and statistical powers ensure the robust analysis, meaning include enough samples to draw valid conclusions. Using biological and technical replicates can account for the data variability and strengthen your conclusions. The choice of sample processing workflows and library preparation protocol is based on the sample type, your research goals, and you should always opt to include controls. Employing the appropriate bioinformatic tools is also key to ensure that your conclusions are meaningful. Cloud-based analysis platforms such as Omics Playground can significantly accelerate the data analysis, and they are suitable for 3' mRNA-Seq and whole transcriptome datasets, guiding your analysis and facilitating your experiments. And with this, we would like to thank you for your attention. If you have any questions or are interested, do contact us and follow us on social media for more information.